Hello, and welcome to Spotlight on Antibodies, a podcast from BioIntron, where we bring you the latest antibody industry trends. I'm your host, Jovina Solistio, and this month in August, we'll highlight drug advances in autoimmune and infectious diseases, as well as next-generation immune cell engagers. Starting off with autoimmune diseases, let's talk Sjogren's. Sjogren's disease, or SJD, is a chronic systemic autoimmune disorder characterized by lymphocytic infiltration of exocrine glands, often accompanied by multi-organ extraglandular involvement. Despite its substantial morbidity, therapeutic interventions have historically been limited to symptomatic management, with no approved disease-modifying treatments. Early biologic interventions, including the anti-CD20 monoclonal antibody rituximab, have failed to meet primary endpoints in pivotal trials, underscoring the heterogeneity of disease mechanisms and the need for more refined therapeutic strategies. Recent advances in our understanding of SJD pathogenesis have renewed interest in targeted antibody-based drugs, particularly agents modulating B and T cell activity, cytokine signaling, and co-stimulatory pathways, reflecting a broader shift from symptomatic relief toward immune modulatory precision medicine. Last month, Novartis made headlines with the announcement of a successful Phase 3 result for two indications of ianalumab. Ianalumab was tested in adults in active Sjogren's disease, and both trials met the primary endpoint of demonstrating statistically significant improvements in disease activity. Ianalumab is a novel, fully human monoclonal antibody with a dual mechanism of action: B-cell depletion via antibody-dependent cellular toxicity, ADCC, and interruption of BAFF-R-mediated signals of B-cell function and survival. Novartis plans to present its data in an upcoming medical congress and submit to health authorities globally, as the drug has the potential to become the first and only targeted treatment approved for patients with Sjogren's disease. This news comes at the heels of Johnson & Johnson's nipocalimab, the first investigational treatment to be granted U.S. FDA Breakthrough Therapy designation and Fast Track designation for the treatment of adults with moderate to severe SJD back in March this year. Nipocalimab is an investigational monoclonal antibody designed to bind with high affinity to block FcRn and reduce levels of circulating immunoglobulin G antibodies potentially without impact on other immune functions. This includes autoantibodies and alloantibodies that underlie multiple conditions across three key segments in the autoantibody space, including rare autoantibody diseases, maternal fetal diseases mediated by maternal alloantibodies, and rheumatic diseases. Blockade of IgG binding to FcRn in the placenta is also believed to limit transplacental transfer of maternal alloantibodies to the fetus. However, it is important to note that antibody treatments for Sjogren's disease has had many more failures than approvals. In February 2025, Kiniksa Pharmaceuticals discontinued SJD prospect abiprubart, an anti-CD40 monoclonal antibody. Earlier in January 2025, Novartis's other candidate, iscalimab, was also discontinued for SJD. Even earlier in April of 2024, Sanofi removed SJD from the list of targeted indications for its CD40L monoclonal antibody, frexalimab, after seeing Phase 2 efficacy data. Meanwhile, Phase 1 and 2 trials for other antibody candidates are currently underway. Otsuka Pharmaceutical Company's sibeprenlimab is currently recruiting for a Phase 2 study. argenx has announced that the FDA has granted efgartigimod Fast Track designation. And Cullinan Therapeutics has also begun a Phase 1 trial of CLN-978, a novel, highly potent CD19 and CD3 bispecific T-cell engager. Moving on to developments for infectious diseases, monoclonal antibodies have emerged as central agents in pandemic preparedness and the treatment of resistant infections. Driven by technological advances in sequencing, display platforms, and artificial intelligence, as well as the urgency of global health crises such as COVID-19, this field is evolving towards a future where antibody therapeutics may be developed in near real time. A recent review highlights the evolution of therapeutic antibodies from the early use of murine-derived monoclonal antibodies to recombinant human antibodies, now central in infectious disease treatment. The review traces landmark approvals, such as palivizumab for RSV and Inmazeb for Ebola, and notes that the majority of mAbs targeting infectious diseases have been approved in the past decade, thus reflecting accelerated innovation. The COVID-19 pandemic showed the use of antibody drugs like bamlanivimab and casirivimab, which were developed at unprecedented speed, even though their efficacy was rapidly diminished by emerging viral variants like Delta and Omicron. To overcome this, strategies like developing multispecific antibodies are being explored. For example, a recent collaboration between CEPI and AstraZeneca aims to develop multispecific VHH antibodies targeting pandemic influenza strains H1, H3, H5, and H7. These camelid-derived nanobodies offer key advantages: smaller molecular size, higher stability, simplified production, and cost-effectiveness. Their thermostability makes them particularly suitable for outbreak responses in resource-limited settings. This collaboration is positioned to expand the utility of VHHs for broader pathogen targets, setting the stage for accessible, scalable antibody interventions during emerging pandemics. Meanwhile, a recent review of monoclonal antibody pharmacology underscores their unique role in personalized medicine. mAbs are now being applied to a set of infectious targets such as RSV, drug-resistant HIV, Clostridium difficile, anthrax, and rabies exposure. These approvals underscore the potential of antibody-based molecules to offer both prophylaxis and therapeutic benefit, particularly in cases where vaccines are unavailable or standard treatments are compromised by resistance or immunodeficiency. Importantly, the study draws attention to respiratory viral infections as high-priority areas due to their global morbidity and mortality. Monoclonal antibodies offer a route to targeted intervention, especially in immunocompromised or pediatric patients, where traditional antivirals or vaccines are less effective. VHH antibodies, due to their small size, high stability, and potential for inhalational delivery, are well suited for neutralizing respiratory RNA viruses like influenza and SARS-CoV-2. Their ability to penetrate sterically shielded viral epitopes and function at mucosal surfaces gives them an edge in early-stage viral neutralization. Now let's talk about some next-generation antibody therapeutics. Immune cell engagers, or ICEs, are engineered molecules that redirect endogenous immune effector cells toward malignant targets by forming synthetic immune synapses. These agents offer a mechanism of tumor cell elimination that is independent of antigen processing and presentation via the major histocompatibility complex. By directly linking immune effector cells, such as T cells, natural killer cells, or phagocytic cells, with tumor-associated antigens on the surface of cancer cells, ICEs bypass several immune evasion strategies frequently observed in tumors, such as MHC downregulation or loss of antigen presentation machinery. Most ICEs are constructed as bispecific antibodies. The resulting artificial immune synapse facilitates targeted immune activation and cytolysis. ICEs represent a scalable, off-the-shelf alternative to personalized adoptive cell therapies. They can be classified based on the immune effector cell population they are designed to engage. T-cell engagers are typically constructed as bispecifics, made up of two single-chain variable fragments, one targeting a tumor-associated antigen and the other targeting an activation receptor on the effector immune cell. They promote the formation of an immunological synapse between T cells and tumor cells, inducing TCR signaling cascades. They also trigger release of cytotoxic granules, leading to tumor cell apoptosis. Meanwhile, natural killer cell engagers, NKCEs, target innate immune NK cells. CD16 is the widely used receptor in NKCEs due to its capacity to trigger antibody-dependent cellular cytotoxicity, ADCC. They often induce cytokine and chemokine production, contributing to immune modulation of the tumor microenvironment. Finally, cytotoxic or phagocytic cell engagers are designed to engage monocytes, macrophages, dendritic cells, or activated neutrophils via CD64, a high-affinity IgG receptor. Bispecific antibodies targeting CD64 and a TAA enable antibody-dependent cellular cytotoxicity and phagocytosis. A recent analysis of 91 research articles published from 2019 to 2025 on immune cell engagers revealed that T-cell engagers dominate this field, comprising of 51.6% of studies. NK cell engagers account for 41.8%, reflecting rising interest in innate immune strategies. Phagocyte cell engagers remain less explored, representing 6.6% of app publications, but indicate emerging therapeutic potential. Innovations in molecular design, such as 1+1, bivalent binding, 2+1, increased avidity, and 1+1+1 trispecific formats, have optimized tumor targeting while reducing systemic toxicity. Current research focuses on improving half-life, minimizing cytokine release syndrome, and expanding the range of targetable antigens through TCR-like modalities. TCE applications are also extending into non-oncologic fields, supported by AI-guided design tools that enhance specificity, stability, and manufacturability. And that's all for this month's antibody industry trends. To learn more insights into antibody drug news, make sure to follow BioIntron on LinkedIn or our website at www.biointron.com. Keep up to date with further episodes from Spotlight on Antibodies by following this podcast.